My name is Maria Gutschi. I'm a pharmacist by training, with a graduate degree at the university at Wayne State University in the US, which I graduated a long time ago. I have spent most of my professional life in a variety of roles, mostly hospital pharmacy, mostly on infectious diseases, but I also worked in government in regulatory role. So that was related to Health Canada. We looked at therapeutic choices and comparison of drugs in order to determine prices, because we have a price control regimen in Canada. I got quite sick, and I was semi-retired in 2019 when the pandemic hit. And I started reading and looking and thinking there's something not quite right with the whole layout of everything. And because of my training, I'd gone to the EMA and looked at their published EPAR, which is the European Public Assessment Report. And when I read that, my jaw fell, because I've never seen a report of that character ever in my 10 years of reading them, where there were so many specific obligations which are required under law to be met, especially for manufacturing. So that started me on this long path, and I got involved with various other groups and worked on the DNA contamination issue, mostly from, helping people understand the regulatory affairs and obtaining vials, which was quite an effort in 2 or 3 years ago. And after that, I'm here.
So, Maria, just stepping back a bit, so would you say what were the main red flags to you? Was it that EPAR report issued by the European Medicines Agency? Because you mentioned something wasn't quite right in the early days.
So it was mostly how the pandemic was reported in the news, where it was, in my view, disjointed. They weren't telling me stuff. It just didn't make sense logically from what I understood how infectious diseases work, because, I worked in a hospital. That was my specialty, was mostly bacterial stuff, not viral, but still I understood. And in my view, the whole thing didn't fit together. It felt like they were running behind an 8 ball, and they didn't know what they were doing, at least the first few months. And then everything settled in. So I just took a step back and said, I'm going to wait. I'm going to read everything. I'm going to figure out what is really going on. So I told, it was funny. And so I did a lot of research in those early days about the virus and about virology in general, to bring up my reading. I feel like I was going back to school, but I'm good at that. I'm good at reading and analyzing a lot of information and picking out what is logical in a logical framework or not. And there was a lot of missing stuff. And when the EPAR came out regarding the vaccine, which of course was my major, given my pharmacy background, it was really, I looked at that, and anyone who reads that would have so many questions that in good faith, they would never take it. So I had told all my healthcare, all my physicians and everything, I said, there's issues with the manufacturing. And they just looked at me like, what's that a problem? I'm like, that is a problem whenever you manufacture a biologic. It is always a problem.
Yeah.
Getting the purity right, always a problem, because the process is the product, right?
Yeah.
The process on how you make a biological is what you end up with. So I told them, until the companies meet the specific obligations, I'm not taking this vaccine. So they backed off, right? I got very little blowback from all the healthcare practitioners I dealt with, because they couldn't understand where I was coming from. So they just let me be.
I see. I
That
see.
was kind of funny.
Were you working,
That
were
was
you
kind of
working
funny.
in the hospital at that time, or you'd already left?
No, I had just stopped hospital work. I was working as a consultant in a small hospital, trying to bring up their antimicrobial utilization, trying to improve safety of drugs and the quality of care given to patients. I had just retired from hospital at that point because I was, for various reasons, mostly I had some illness I had to deal with and some family issues.
Yeah. So for people who don't understand obviously what an EPAR is, could you just break down what that acronym stands for?
It's the European Public Assessment Report, and it's a report all regulatory bodies, like medical regulatory bodies like the EMA, FDA, Health Canada, TGA, all do a summary of all the data that's been submitted for them in order to be authorized for sale and marketing of a drug. And in that is usually mostly heavy on the clinical things while they assess the clinical trials, and they do give a summary of the non-clinical data that used to support the submission, and manufacturing is usually quite tiny, right?
Yeah.
It's usually quite tiny. And then afterwards, they make an assessment and say, this looks good, but you are still required to provide this and this information, depending on how
And
advanced the drug is.
just to stop
Yeah,
you, and
that's
those would come under, and correct me if I'm wrong, Maria, would be the specific obligations that the marketing authorization
yeah,
holder needs to meet. Right. Okay. And would you say that
correct.
yeah,
So there's actually two. There's specific obligations which are required under, for the EMA is required under law to meet within a certain time period, usually a year. And then there are a bunch of recommendations. They don't have
Maria, you're cutting out.
uh,
Maria, you're stalling.
they don't have
Sorry, I
yeah.
in there it just all went weird. Okay, so I'm gonna obviously edit that, but you started to stall. I don't know if it was from my end or your end. So if it happens again, I'm gonna switch to my hotspot and go on to 4G. I'm not sure what. So I would like you to start again when I asked you about the specific obligations, and then it all went a bit funny. So just can you remember the question I asked you? It was
Yes, but just explain specific obligations, right?
yeah.
So specific obligations are requirements under regulatory authorization that the sponsor, i.e. the drug company, must meet within a certain time period or risk their authorization. So it is has the force of law behind that. You have to meet these specific obligations, which are usually extra data or extra studies that are required in order to keep your authorization going. So the EMA had come up with five, of which four of five, six of them, of which five were manufacturing. Normally, specific obligations deal with clinical outcomes. You need a trial in this, or we need the 6 month data, or whatever.
I
But
get
these were on manufacturing.
yeah.
And yeah,
So that would be, you say, would be
so
highly,
that was what was unusual.
yeah, okay. Right, that's very interesting. And would you say also why was the EMA's version, because obviously all the regulators did this, FDA, I'm assuming MHRA, was the EMA's one the most transparent and easily accessible for the public to get? Because why did you zone in on the EMA's?
Uh, that's a good question. Yeah, and the answer is yes. It was the most complete, most thorough, and most transparent. I think it's maybe a surprise for people for me to say that, but I have found that the EMA reports are really well structured, are detailed, and I've been going to them for quite some time as my primary source for review rather than the Canadian or the FDA ones, which are quite jumbled in my view and not quite as transparent. So these have been, the EPARs have been my go-to for my assessment of drugs when I worked for government.
And
So that it was my standard.
Could you give me, sorry to interrupt, Marie, could you give me, when the alarm bells started going off, when you were looking at the EPAR report, obviously this is, can you tell me the time period? What was the date?
February 2021.
Right. Okay. So that would have been at the
right soon
start.
after it came out. Yeah.
Yes, exactly.
Yeah.
So the start of the vaccine rollout, and that's when to you the alarm bells started to go off when you were reading these reports. Great. So obviously you said you've been very specific where there were big
Yes.
issues with
Yes.
manufacturing, right? With the quality control. Now, fast forward to where we are now, quite a few years later, and now we're seeing the aftermath of that, aren't we, Maria? I mean, please, could you speak to what has been found in these vials? And then we can then speak to this Canadian open letter that I know you've headed up that's gone out to all the Canadian government representatives and health officials.
So pretty well, all the issues, most of the issues that were identified in specific obligations are still a problem, right? They are still a problem.
Yeah.
And this is, so the EMA did a very good job identifying these issues, and they're still a problem. And of course, one of the big problems that they identified was what exactly did this product make? Did it actually make an authentic spike protein? And then they talked about all the potential contaminations, of which one was double-stranded RNA, which nobody talks about. And DNA wasn't a huge specific obligation in there, but it was one of the recommendations that was also a concern. It wasn't as much, but what we found is that when Kevin McKernan in April 2023 first published on these things. And so I said, aha, we should do in Canada issues. And so I managed to get a bunch of vials from a very sympathetic pharmacist that worked in community and managed to find David Speaker to analyze them using Kevin's kits. And we found the same effect. And I was able to get a whole lot more vials. And that, of course, that started us down this pathway on exactly what's happening at the manufacturing level, why this is happening, and everything has ballooned out from there. And as we see, the more we look,
Maria, because it's a very important paper, because this is to do with a paper that was published, right? That you were you involved in that paper that was published with David Speaker, and was it David Wiseman was involved? Could you
Kevin and
yes, Kevin McKernan, could you just, what was the title of your paper and where did it get published?
oh my goodness, DNA fragments found in Mrna and Pfizer in Ontario, I think is kind of the short form. I can't remember and all. It's a preprint. It hasn't, so I don't know what the long-term outcome of the actual paper, but nonetheless, it basically verified what Kevin McKernan found with vials that did not keep chain of custody, but because I know exactly where they come, how they come, and they were closed, they were new vials, we were able to at least address that issue as a problem. And from then on, other labs have done similar things. So there's quite a few labs that have verified the amounts of DNA that is in the vials.
Can you remember the finding for your paper? What was the results? What was the it was how many from what you were expecting to what you actually found? What was the sort of level of the residual plasmid DNA?
Okay, so the residual plasmid DNA, one of the ongoing issues is how you measure it. So in the EPAR, or the standard way of measuring it, was with quantitative PCR. So that was done using quantitative PCR with the kits we have, all the vials met the guidelines, the WHO guidelines of 10 nanograms per mil, or per dose, I should say, but using fluorometry, which measures the smaller DNA fragments, then all of them had quite a lot of DNA. Contrary to what people expect, Moderna had more of the smaller fragments and actually had more total DNA in the vials than Pfizer did. Pfizer had bigger pieces. And so that was a concern because the plasmid, the DNA in these vials have regulatory sequences. They are plasmid DNA. So they have potential action within the body, as opposed to the DNA that might be found in other products. In addition, of course, we have the lipid nanoparticles, which transfect the lipid bilayer, allowing those levels of DNA to enter into the cytosol. That is a game changer, right? That is the problem with the DNA in these vials. And secondarily, and probably most important, or even more important than the quantity, it showed the variability in the DNA from one lot to another.
Right.
Okay, especially for Pfizer. That is something that does not normally happen in biological manufacturing. It's a big no-no. Variability is a problem because it's better to have higher DNA levels and be standardized, because then you can work to bring everything down. But the variability in the DNA is still there even today. And this speaks to really huge issues in the whole manufacturing process of these products.
Right, I see. Okay. I just wanted to, because you first said when you did this, you're talking about how that the standard measure for the residual plasmid DNA, you said in the beginning it did meet the WHO guidelines. As it was that for the bigger particles? What could you just, but you found lots of.
So So the standard is basically using qPCR. It's a total of 10 nanograms per mil. For DNA vaccines, we also have an extra standard that all of those pieces should be under 200 base pairs. When you have it under 200 base pairs, it's less likely to be a complete gene and less likely to integrate or cause a problem in the human body. So you have basically two things, right? You're looking at the amount and the size. But the size of the DNA fragments are not measured as part of what's called the critical quality attribute. So it's not required to be measured as the standards used to determine quality control for these products. So with using certain techniques like fluorometry, you can actually determine what the size, or nanopore, I should say, you can determine the sizes of these fragments. The issue here about the smaller fragments, which was thought to be not such a problem, is even small fragments can act upon other DNA, that is, have regulatory or have action upon the genome.
Could you just explain to the listeners and the viewers the issue with the Blotgate scandal, which I also have investigated myself a while ago, and how that pertains to the specific obligations that BioNTech was supposed to meet for the European Medicines Agency?
So you bring up a really important issue, and I really appreciate your reporting on Blockgate. And anyone reading should really, really look at what you uncovered, what was done. What's interesting, what Blockgate was all about is one of the. I'm going to step back a little bit. We have for this product, and this is, you have to have. How do you know that the product, that this vaccine, quote unquote, is doing what it's supposed to do? How do you know what are the quality parts, what parts of the vaccine need to be at high quality in order to get safety and efficacy. So one of the things you need to find out is what exactly, like how is the vaccine making the spike protein, and what does that spike protein look like? Because after all, this is what we call a prodrug, right? We are not giving the active ingredient as opposed to every other vaccine. We don't give the antigen. The antigen is the spike protein, but the ribosomes, your body has to actually produce that from the mRNA or the blueprint. And we need verification that that blueprint is making the correct spike protein as it has been, as the blueprint says it does. And what Blockade said is that, well, there is some concern that that is not occurring. It was bigger and bigger than what they expected. The BioNTech said, oh, it just has a bunch of sugars on it. You know, that's why it's bigger. And the EMA say, oh, that's fine. Get rid of the sugars and redo this over again. And to the best of our knowledge, we're not sure that that data has been completed entirely, because not only does it have to do for a standard, what they call a performance lot, but the EMA wanted to do it for several lots to see if there's consistency in the spike protein being made. To the best of my knowledge, that specific obligation hasn't been fully met. Is that that's my understanding?
Yeah, well, this is insane. So this is 2025, and to this day, it still hasn't been met, this obligation. And this is to do with the unusual weight size, measured in kilodaltons, if I remember, which showed up
Correct.
on these Western blots. And BioNTech just dismissed it as sugars, and the regulator, the EMA, was okay with that. Yeah, it's
As
really.
far as I'm concerned. And then this brings it up to the ribosomal frame shifting paper,
Yes. Yes.
correct?
Just that's quite a big term. So people who are not scientific, please explain in very simple terms what that means.
Well, join the club. If you told me I would know what ribosomal frame shifting was 3 years ago, I would have said you were nuts. It is really because the mRNA is a bit of a blueprint, almost like a computer code. As it goes through the ribosome, it tells the body how to make an amino acid, right? So there's three codons that, or three little pieces of info that says form this protein. And it goes through the ribosome, but on occasionally it'll shift. Okay, so instead of it going 1, 2, 3, 4, it'll shift and go 2, 3, 4, 5. And when that happens, the proteins are not made in the order that the blueprint says it should be made, so that you have small changes in the protein. This is really important because small changes in protein can change toxicity, efficacy. It affects how things that people don't talk about, how the sugars apply to the spike protein, because this spike protein is a glycosylated spike protein. And when the sugars go onto the protein, they dictate where your antibodies are formed. Okay, this is really important. So if you have a change in those amino acids, you may change how the body sees this spike protein. It may be more toxic. It may be ineffective. We don't know. So that's why the ribosomal frame shifting is actually quite important, and in some ways was predicted by the EMA in their original review. And that's what they wanted to see.
Yeah. And could you also speak to that landmark Cambridge study that published
Right.
about this ribosome, which actually did get picked up in the mainstream media a little bit. And I know it was dismissed as well as not being very important, or it doesn't matter. But there was this big they discovered this ribosomal frame shifting, and maybe this misfolded proteins. Maybe you could speak.
Yes. Well, that goes to the fact that the proteins are different, so the folding gets different, right? So if they're not in the right order, the folding therefore changes the glycosylation. This was a paper published in December 2024 in, no, 2020,
2023, I think.
Three,
Yeah.
correct. Oh, wow. Yeah, December 2023 by Mulrooney et al. It took about a year to be published. And if you read it, yes, it is semi-dismissed, but really when you know about this kind of potential problem, it is not something that really should be dismissed and more work needs to be done on it. I don't know if you're aware, but I was at a conference in Ottawa called the Ribo Club, where all the RNA scientists throughout Canada, and actually there was some Nobel winners who presented at this, and I presented a poster on this stuff. And I talked about the ribosomal frame shifting, and I had a lot of the RNA researchers saying, oh, our body has mechanisms to get rid of this. And I said, fine, where's the empiric evidence for this? We can't just say, oh, this should be taken care of if we don't have the empiric evidence to say, okay, this is not a problem. So that's what we need. We've identified the issue. We need the empiric evidence to say, studies to say that this is the biggest risk is autoimmunity with this,
Yes,
right?
yes.
That's the
Because
biggest
I
risk.
remember the paper even speaking to picking up on the aberrant proteins being produced, the off-target proteins, and obviously the mystery of not knowing what are these proteins being produced other than the spike protein. And now you obviously, yes, I mean, autoimmune is a huge issue, which the EPA report did speak to many years ago. They were worried, the EMA was worried about the autoimmune diseases. But did nothing about it, right? To this.
Yes, or asked and was ignored by the biotech and then not followed up, right? There was no real follow-up with teeth to make this happen. And given BioNTech and Pfizer's abilities and money and labs that they have, this would not be difficult to achieve in a short period of time, okay? Would
Yeah,
not be difficult because we're not asking for randomized controlled trials or that kind of thing. This is primarily a lab-based kind of result. And there really is, shouldn't be that taken long for this kind of answer to be determined,
right.
in
So
my view.
we are in 2025. It's almost 5 years on. And we have a cascade of obviously we have excess deaths happening. We've got skyrocketing cancer rates. Um, we have a host of autoimmune diseases, breaking out as well. I mean, um, Maria, what could you say? I mean, do you blame the COVID, well, we can't really say vaccines, but these biological products? Do you lay it down at the feet of the COVID shots?
So I am of the view that the virus itself is not benign. Okay,
Yeah,
I'll put that straight out. I do not think it's just a cold. I think it has a variety of activity. However, I do think the vaccine at least magnifies all those adverse events, and if not a primary mover of many of these adverse events. So what this is the biggest issue when you have a new disease. Let's just say that this is novel, and you're developing a new vaccine. So, and you have the data that is so mixed up because you do not know the natural history of COVID and you don't know the long-term adverse events of the vaccine. So what we have is a mess, and we need research to straighten this all out. I am of the opinion, of course, based on the issues that were raised actually from the EMA and the overall knowledge of how this mRNA works, that the majority of these adverse events are from the vaccine. But we need the research, and we need the ability to be able to determine that. And most importantly, and if we can't determine that, we can't find treatments or something to help patients who have these adverse events, right?
yeah,
So if we don't understand this all, we can't help them.
yeah, yeah.
Yeah.
Going back
So
to the contamination scandal, though, Maria, this is the SV40 promoter enhancer DNA sequences that were found, I think, primarily just in the Pfizer BioNTech vaccines. I know residual plasmid DNA
yes.
was found in both Moderna and Pfizer's, but can you speak to that and the link to cancers?
So Mrna did the sequencing of the vials was the determination of the SV40, finding that. Because as we all know from previous polio vaccines, the entire SV40 was contaminated polio vaccines and is a known oncogenic virus. Now, what Kevin found wasn't the entire virus. He just found it's about 350 base pairs, but of that, there's a promoter, which overlaps with enhancer and has the origin of replication. The origin of replication is important because it allows this section, this bit of DNA, to multiply. The actual promoter enhancer is 72 base pairs. So that's under the 200 base pairs that we thought was safe, right? So that even though you may be breaking up the DNA and you're not measuring the quantity of SV40 if they're under 200 base pair by quantitative PCR.
Can you just
Is
speak a
that
bit?
clear?
Can you
Right?
just
So
speak
that
I just want to stop you there. Can you speak back to the origin of replication? Was it Ori you said? ORI?
Ori, yeah,
Yeah.
yeah. So you've
What does that
got the
stand for
promoter,
again?
which promotes. So there's this SV40 sequences that were found in the vial contains a promoter. A promoter can promote a gene that's in front of it or behind it because it's bidirectional. Then you have an enhancer, which kind of overlaps. The enhancer enhances genes further down. It doesn't have to be beside it. It enhances genes a few hundred kilobases away, which I find absolutely fascinating. And then you have a section called the ORI, which allows that bit of sequences to replicate.
Okay.
So right? Does that make sense?
Which is
So
a
you
big
have all
problem.
of that. It's a huge problem. It's a huge problem. And the SV40 has this, is the most active part. These sequences are the most active part of the entire virus. So, which if it gets integrated into your genome can and is in front of an oncogene, can promote the oncogene, or it can enhance the production of it, and it can replicate itself. Not only that, it can drag other DNA into the nucleus because it has that ability, and it also blocks something called P53, known as the guardian of the genome. So it has a lot of potentially proto-oncogenic activity.
Just to stop you there,
That
Priya, just to stop you there. So the P53 gene, which is really important, so that suppresses, that's our natural cancer suppressor gene, right?
correct? Yeah,
Okay,
yeah.
so the fact that this sequence found in the vials blocks P53 is a huge red flag, a huge problem.
Yes. Now, that doesn't mean that that it's the only cause, because cancer is quite complex to happen. But as I've said for a long, long time, from a regulatory point of view, okay, you don't want any SV40 sequences in any vaccine. And in fact, in these FOIA requests that we have examined, Health Canada admits there are no SV40 sequences in any vaccine that is approved in Canada. So if
But they, hang on,
so,
stop it. So, but they did admit that there was residual DNA, right?
that's correct.
But
Yes,
they
and
have
now
not admitted that there's SV40 sequences found.
yes,
Okay.
health. Yeah, health. Yes, that there were no SV4 sequences in any other vaccine. So the fact that they're found in Pfizer was a huge red flag, right?
Okay, so they admit
And
that
I
it's
give,
found in Pfizer.
yeah,
Okay.
Health Canada was the first regulators to admit that that was the case. I give, some credit to those regulators. They were absolutely horrified, which you can tell from some of the FOIAs, which I have put in the letter. And so the SV40 is a ongoing concern. On top of that, it was not disclosed to the regulators. To me, this is absolutely unfathomable. Okay? Absolutely unfathomable, because this should never happen. Even let's pretend that Pfizer says, I need the SV40 in order for the plasmid to work, whatever. Health Canada would say, okay, are you sure? Can it be done without? Because Mrna has it done without. If you think you can't do it, then your job is to remove every single last bit of it, because we don't want any of it in any of our vaccines. But what Pfizer did not disclose that to the regulator. And this is an outright lie and what I call hard fraud.
Yeah.
But they do a lot of what I call soft fraud, which is if you're smart enough, you can figure it out. But hard fraud is something like this, a right out lie, a right out undisclosure of significant active
Yeah. Yeah, it's
DNA. Yes.
appalling. It's so shocking. So Health Canada, you're saying from these FOIA requests, they are shocked at finding it. They didn't expect it, but they haven't done anything about it. They have not halted. They haven't recalled. Did they even
Yes.
tell Pfizer to sort of clean up their act and remove it, or
They
what?
did. They
They,
actually did.
oh,
Yes. So the very next day, Health Canada did what is called an issue analysis summary. An issue analysis summary done in regulatory fields towards the end with a goal of changing the label. In other words, who would get it, or removal from the market. So they had actually started that process and it was quashed. We found that out through some parliamentary questions that we asked.
hold that right there,
And
because this is really huge. So you're saying they were in the process of fixing it, right? Health Canada was on it, they were going to get it done, and then their own investigation was squashed from within.
yeah,
Wow.
that's what it appears to us. Yes. And then they asked, they had a meeting with the FDA and the EMA. They revealed this. They wanted to know what can be done. The EMA took a bit and went on board. So Health Canada actually asked for an explanation for the SV40. They asked for what the average size and whether, how much DNA was under 200 kilobases. And they asked for the whole justification and whether or not this was integrated into bacteria, for example. And that's a 56-page report that we asked for. That's a colleague called Scoops Magoo, who's really good at doing ATIPs. And we got it back mostly redacted. So we've asked the Information Commissioner of Canada to unredact this 56-page report that Health Canada asked of Pfizer. And the Information Commissioner actually took our complaint and asked for it to be unredacted. So far, Health Canada has still stalled on that. And so we're working on trying to get that report unredacted in the interest of public.
But you're saying even that report, even if you get it unredacted, so it's obviously it's specifying to what they need to do to fight, how to clean. But you're saying that never went anywhere, that they never enforced it, really. They never followed up even on their own report.
Yes, or whatever the report was. I mean, the FOIA says Pfizer told Health Canada, like, nobody else is asking us why we need to do all this, so we don't feel like we need to answer your questions.
I see.
Then they give them, which is actually not unusual. That happens a lot. And then they give this report that justifies everything. Since we don't know what is in the report, we don't know if that was justification enough for Health Canada or not. And not only that, it appears that the other regulators also got something similar. That's what we think is happening. So again, it's not as if the regulators don't feel that this is important, but somehow this request to remove it was made by Health Canada. But as far as I know, the SV40 segments are still contaminating the vaccines as of 2025.
So essentially, Pfizer just ignored it, really. They've just
Correct.
ignored,
Correct.
and Health
Yeah.
Canada going, okay, that's fine.
Yes.
Is
EMA
any
going okay,
evidence?
the FDA going okay. Correct.
Yeah.
TGA. Like, I do not understand this. And that's one of the things that we bring up in our letter, right?
Yeah, we will speak now to your letter. Please, Maria. Tell me about this open letter uh, that was issued recently and how you were involved in this.
So we decided to do an open letter because there were several that were, happening in the EMA, the North letter in the European countries, and we had stuff happening in Australia. And I wanted to write one that was focused primarily on the Canadian issues because of all these FOIAs or ATIPs that we had that revealed a bunch of information that is not normally known. So we were focusing our letter specifically on what was identified by Health Canada scientists themselves as an issue. So the DNA contamination was one. They're not Health Canada scientists were not that concerned with the quantity, but they were concerned about the variability. So that was one thing. They were concerned about the SV40. And what we mention is even if we think the SV40 segments or sequences are of no specific, toxicological issue, by not revealing it as required by the WHO guidelines that were written in 2022 or drafted in 2021 that says every single part of the plasmid must be reported, must be disclosed to the regulator. They are not meeting even the WHO guidelines, let alone FDA or other ones. And that in and of itself is a problem that needs to be addressed. We also say that the lipid nanoparticles and the issue of transfection is something that needs to be looked at specifically and independently from all these other issues. And the other two things that came up was the ribosomal frame shifting we talked earlier, and shift to the IgG4, which is the tolerating immunoglobulins. Now, this is not part of my expertise, so I don't really fully understand this, other than it is more likely for you to tolerate an infection from COVID. And this was identified as a potential issue by Health Canada scientists themselves, and kind of just like, oh, this might be a problem, but we're not really seeing it, are we? That kind of response. So we wanted to bring all these issues forward and allow the health science, the regulatory bodies themselves to say, yes, we've identified these issues and we have tried to address them. And I wanted to give them an off-ramp, if I could, to say, yeah, we agree with you. Okay, can we work together at figuring out what to do about it and how to resolve these issues and how to study them? So we are asking for, a review of the regulatory authorities, the risk of cancer, the contamination, and the fact that and the adverse events occurring as a result of it. This is my goal, right? To try to identify that we have the same concerns as many of those scientists themselves do.
Yeah.
I don't know if we succeeded,
Yeah.
but yes.
Yeah. So when was this letter sent out? Was it end of February?
Yes, the beginning of March, we've sent it out. We're collecting signatures and we're planning to send it to all the MPs, the senators. But one other thing that we're planning to send it to is all the chief medical officers of health in Canada, which are the provincials, since we don't have a national medical act. We have it for each province, so we have to send it to each provincial officer of health. I am fairly certain that most of these public health officials in the provinces have no idea that there's SV40 in the vaccines. I am of that opinion, and they need to know. I
Yeah.
don't think this data has been shared with any of it's not that the Public Health Agency of Canada, which is similar to the CDC, does not know. We have evidence that they do, but I'm not certain that the remainder of the actual people doing the work at the at the provincial level do. So that's one thing that I
Could
was
you just
hoping to
speak a
achieve.
little bit to do with the transfection? And this is to do with the lipid nanoparticles, which we know the modified mRNA is encased in these lipid nanoparticles. It can get into the cell membrane. It goes, it increases the transfection rate. But can we talk a bit about DNA integration, how it can, that foreign
Oh,
DNA integrating into, the human genome is there, would you care to speak a bit about that and what Philip Buckholtz has found as well in the US in his lab?
I'm not sure. I do know that he's found a fair amount of DNA within
He
within the vials.
Yes, I think
Yes, I'm not
he proved it in vivo,
familiar.
no, in vitro about but not in vivo, but in vitro in the test tube. But what are the dangers of we have foreign DNA in these vials, in these injections? They're encased in these lipid
Yeah.
nanoparticles. They travel through, cross the membrane. What's the problem with that? What's the red flag?
So the lipid nanoparticles in and of themselves are inherently dangerous for several reasons. And one of the things that had bothered me with the EPAR when I first read it was that the lipid nanoparticles were seen as excipients. That's how they were regulated. Excipients are products that do not have pharmacological activity on their own. And that is not true for the lipid nanoparticles. So that is something else that needs to be addressed.
Yeah.
These are nanoparticles. Nanoparticles have actions that are much different than single molecules or molecules in a bigger state. Because they're nanoparticles, they're very reactive. The outside is much bigger, the surface area is much bigger than the mass inside. So they're extremely reactive, which is what is used in order for them to basically cross the bilayer of the cell. And it does that without using receptors. Most drugs, in fact, everything I know uses some kind of receptor or ligand to get into the cell, which can therefore be modulated, upregulated, downregulated, that kind of thing. But the lipid nanoparticles are like a knife or like a bomb that goes into the cell membrane and just brings everything that's inside or on the lipid nanoparticle into the cell. Okay. They are about the size of a virus, and they are like a bomb that just hits the the bilayer, and just gets engulfed totally and brought into the cell. So everything that's in the lipid nanoparticle is now in the cytosol. And that includes any contaminants in the lipids themselves, which we know had been, that this has been cleaned up, but included metals like arsenic, antimony, chromium, that would be in there. Any leftover parts like the double-stranded RNA that was made with the mRNA, that was part of the process of making the mRNA, including enzymes that were used. That's also going to be inside the lipid nanoparticles. That's inside the cytosol. And then we have the DNA, the mRNA, and anything else. All of that goes into the cytosol. This is something the cell is not used to because it doesn't go through a ligand or through a receptor. So it is basic. I like to think of it, I used to think of it as like this whole army that came in like a Trojan horse, and all of a sudden the horse is opened up and you got, all this army in your body, in your cell, that the cell was not ready for.
And
That's the way I
what
see it.
about the
And
nucleus of the cell? What about the DNA going into
so,
the nucleus?
yes. So the DNA in the cytosol itself is a problem, and it can stimulate other pathways that are detrimental, or um, um, and certain depending on the quantity and because of the SV40 and even that's, I think, what Philip Buchholz said. When you have a ton of buckshot of DNA, the chances of the DNA, even without SV40 there, to get into the nucleus and therefore be taken up by the genome, or even stay in the nucleus and not get taken up by the genome, but stay in there and produce proteins or produce or affect the genome is very high. Just because of the nature that you've brought the amount of DNA 100 to 10 times over what would happen if it was still sitting in your serum, for which the body has a multiplicity of actions in which to degrade the DNA that you may find within your serum. The lipid nanoparticles basically protects it from all that and has action within the cell that is detrimental in and of themselves.
Yeah. I mean, it's just like a whole host of alarm bells, isn't it, Maria? I mean, it's just essential things that, and then we are seeing. I mean, obviously, you've heard of Professor Angus Dalgleish in the UK, he's managed to prove it's with the repeat injections, the risk of cancer going up with the boosters. He's done studies on that in the UK. So it is very, very alarming. I mean, have you had any response from any of the government reps, public health, any from this open letter so far?
And so far, well, it hasn't been officially gone to them yet. We're still collecting things. But people had sent letters to Health Canada and the Public Health Agency of Canada in the past. Other groups have, and no answer. It basically goes into a void. I also want to mention that we are partnering with Quebec, Info Quebec, a very active group that has done quite a bit within Quebec, which was one of the jurisdictions in the world that had the worst lockdowns and the worst mandates. And it's a very well organized resistance in Quebec. So we're very happy to have that as well in this letter to address Health Canada. And public health, actually, PHAC, our public health, to me, is the biggest impediment to actually looking at this vaccine in a sober, logical manner, if you know what I mean. There is one thing I would like to bring up that maybe not be related. One of the things that bothers me a lot is that when we address adverse drug reactions, because under law and regulation, these are identified as vaccines instead of what they really are, which is a drug or a gene therapy product. Because they're identified as a vaccine, we're using what's known as the Brighton Collaboration Causality Assessment for adverse events due to vaccines. Yeah. This Brighton collaboration does not address adverse events like it does for drugs. It doesn't use a probability assessment, right? It doesn't take into account if you've got heart failure or if you have autoimmune disease, that giving this may be a higher risk than not.
If you have
the causality, yes, and so it's extremely hard to show adverse events using for a product that doesn't act like a vaccine, whose mechanism of action is not a vaccine, yet they use an adverse event assessment rubric that does not apply. And I think this is something that really also needs to be looked at. Yeah. So that's my little plug for that. Okay.
Yeah. No, that's brilliant. I mean, obviously, your main message is what, Maria, is that these shots need to be immediate halt. They need to be recalled, investigated. If I've left out anything, please step in.
And studied, and basically do some hard work, some funding to look at the mechanisms, to look at not just epidemiological, some bench chemistry, some lab-based studies to start, and then we can work on how these products actually work in the body, what they actually do, and so that we could start looking to therapies that are going to be that may make a difference.
Yeah.
That is the goal. So it's not only like recall them, that we have enough evidence, we feel at this point, to show that the risks do not outweigh the benefits, number one. Number two, that the contamination, particularly with the SV40, is a risk too high, that they should no longer be considered vaccines and taken off the market. And thirdly, that we have the research done to actually look at these in detail in a objective manner, together with the virus itself.
Yeah. That's brilliant. Well, thank you so much, Maria, for taking the time to speak with me and for all your independent work and research. I really thank you so much. If there's anything you want to add, please feel free before we end the interview.
Thank you very much for interviewing me. That was sometimes you feel like all the work you do is all in the background, which is my default setting, so to speak.
Yeah.